Good morning, good afternoon, good evening. I remain your resource person, Wokike. Please make sure you subscribe as you're watching this video. Today's topic is still going to emphasize on our normal neuroscience of psychiatry aspect of my topics or my courses. I've told you before that the neuroscience of psychiatry is the major, most critical aspect of psychiatry that need an in-depth concentration, because it's through this neuroscience aspect of psychiatry that we demystify and at the same time understand how the external environment relates with the internal environment, how our body responds to external stimulus, how those responses are being translated or transduced into electrical mechanism known as action potential before it will now undergo some release of neurotransmitter by the presynaptic terminals bundle. The presynaptic terminal bundle houses the neurotransmitters that can only be released by the stimulation of a particular neurotransmitters that is related to the external stimulation. And I say that these particular sensations can as well result to awareness and perception, which is just like a psychological knowledge about what is happening or psychological response to external environment. That is where feeling and experience occur. This scenario have also been illustrated when we are talking about the chemical substances that are responsible for this transmission of impulses from the presynaptic part of the neurons to the postsynaptic aspect of the neurons, because of some essential processes that take place, which we are going to discuss today. Before a particular sensation will stimulate a neurotransmitter, for that neurotransmitter to influence the postsynaptic receptor sites in the postsynaptic neuron, that particular neurotransmitter must be in an amount that it can influence that neurotransmitter. So the minimum amount of a released neurotransmitter within the synaptic cleft, so that it can have the ability or the tendency or the pressure, or let me call it the awareness, or have the intensity to stimulate the postsynaptic receptor site, is called absolute threshold. Then sometimes there are some response that occur unaware, and most of these response occur as a result of subliminal stimulation or responses, which is the lower amount of a neurotransmitters released in the synaptic cleft that is not as concentrated or as full or as many to that of absolute stimulation. So we are trying to tell you that for you to have an absolute threshold, that means the action potential stimulation that will result to influx of a desired ion can be positive ion. Then when it is positive ion, remember there will be a depolarization of that cell, which will now lead to release of a neurotransmitter that is responsible. And this neurotransmitter that is responsible can be an excitatory neurotransmitter because it is a positive ion. Hence, it will now potentiate a process known as action potential. And for this action potential to take place, that means that receptive impulse or the impulse that is being received from external environment have translated to different ion which is desired in order to stimulate the interstitial and the extrastitial aspect of the neurotransmitter, leading to leading to exchange of ions, hence resulting to action potential. And for this action potential to take place, that means there will be release of neurotransmitters in the synaptic left. And this neurotransmitter that is released in the synaptic left will be to an amount which it can have the capacity or the ability to influence the postsynaptic receptor sites. And for this minimal amount of these neurotransmitters found in the synaptic left that have the ability to influence the postsynaptic neuron is called the absolute threshold of a neuron. Then below absolute threshold, which is lower than the amount that we release, which an individual may not respond to it because we will not take cognizance of that particular responses or sensations. We say that particular stimulation is known as subliminal response or stimulation. So these are so many things that occur within the release of neurotransmitters. I remember I told you before that major neurotransmitters are found or located or produced in the cell body, where the minor neurotransmitters are produced or secreted or synthesized in the axon or the synaptic terminal. So this also affects the release of these neurotransmitters. So if there is a minimal, let me say subliminal stimulation in the neuron, there is possibility of the release of those minor neurotransmitters that is found in the synaptic left, in the synaptic terminal and that of axon, because it is closer to the bundle and that of the synaptic cleft. Well, there must be a concentration of those neurotransmitters, concentration of that action potential to initiate the release of major neurotransmitters. And for this to occur is because of the position of where this neurotransmitter is found. And where is that position? In the cell body. And the cell body is far, very, very distal from the synaptic cleft or terminal vesicles or terminal bundles or synaptic terminal. Depends on a particular source. So this process of action potential stimulation that will lead to release of neurotransmitter are more of electrical transmissions. Hence, transmissions is divided into two: electrical transmission and the what? Chemical transmission. So chemical transmission is start from where electrical transmission stop. This one occur as a result of the released neurotransmitters that have the ability to be influenced by the postsynaptic neuron. So that ligation or for a particular neurotransmitter to identify its receptor site in the postsynaptic cleft, leading to some chemical what ligation by some ion channel ion gated ion, like there are some what we call ligate ion and the protein pump or protein ligands. Some protein pump receptor pathway enhance or potentiate the attachment of a specific neuron or transmission of that neuron to the postsynaptic cell. So some neurotransmitters are potentiated or enhanced by ion ligated what pathways. And for this action to take place, it must be that identical neurotransmitter that is released must have that specific response to its receptor site in the postsynaptic neuron. What I mean is this: for a neurotransmitter to be released by exocytosis, the neurotransmitters that is found in the synaptic what vesicle or terminal bundles or synaptic terminals have its receptor site in the postsynaptic neuron, and these neurotransmitters does not go into another site. Dopamine discover dopamine receptor pathway or SAT, and it initiates what the transmission, which will now result to the brain getting aware of that particular perception or sensation. If it is that of GABA, so also GABA receptor sites will now open and receive the impulses. Same to every other neurotransmitters. That is what I mean. So the specificity of a neurotransmitter determine the receptor site where it can influence, and this must be in an amount that this influential aspect of these neurons can activate the presynaptic or influence the activity of the postsynaptic neurons. So this is what takes place for a particular transmission of impulses. I want you to have this short note like this. If you're not understanding, listen again for recap. I said in sensation and perception, there must be so many responses that take place, which will now lead to a release of a neurotransmitter. And that neurotransmitter that is released have already been stored in the terminal bundles. So because of stimulation by the external environment can lead to release of a particular neurotransmitter. For instance, if it is excitatory neurotransmitter, there will be influx of calcium, which will now lead to depolarization of the cell. And when the cell is being depolarized, there will be influx and exflux, influx of positive ion and exflux of negative ion, hence resulting to a process called action potential. And this action potential processes is otherwise refers to transduction in sensation stages. So for this to take place, that means there is a possibility of exocytosis to take place. Exocytosis is the release of matured neurotransmitters that is already found in the synaptic terminal bundle. And when it is released, it found its way in the synaptic cleft. And this synaptic cleft is where it undergoes process of filtering, identification, and discovering of its own receptor sites. Remember, we have presynaptic and postsynaptic neuron, and the presynaptic neuron is the one that is producing, is that particular neuron that is responsible for the production or synthesis, at the same time transmission of those neurotransmitters to the postsynaptic. So the postsynaptic neuron have some receptor sites that are specific to the neurons that will be released, to the neurotransmitter that will be released. So if, for instance, they say we have 100 billion cells, so also we also have 400 million receptor sites. I mean 100 million receptor sites. And this 100 million receptor site is also equal to the amount of neurotransmitters that might also release. So for instance, we have GABA being the highest in the brain, followed by, the glutamate being the highest in the brain, followed by GABA. Then before you will now talk about other neurotransmitters like dopamine that have up to 500 what million. So for that cause, I want to tell us that this process of action potential, sensation, translation, transduction, and the stimulation of the cells causing the release of neurotransmitter is called electrical what? Transmission. Where the chemical transmission is the ability of those neurotransmitters to identify each receptor site in the postsynaptic neuron. And the discovery of this will now lead to enhancement of this thing by some ion ligate or ion channel ligated ions or cascode, and that of the protein pump activators. So most of these proteins or ions enhance the reception of each of the different neurotransmitter depends on the one that is released to instigate or incite neuronal changes. And when there is this stimulation, or let me say identification and the incite or and the excitation of the postsynaptic neuron, these neurotransmitters undergoes some series of characteristics. They either die off, they either undergo reuptake mechanism, or they decarbonize. Can also be damaged by the enzymes that is responsible for the neurotransmitters. So I can also go back, which is not clear in science. In a nutshell, I want to tell us that we are going to end here for now, so we can go into another topic. Follow, make sure you template, and as you template, you will see more of my video. Thanks.